Okay, so this can be more beautifully explained by using this diagram. See the stem cell hierarchy. You can see the different colors here. This color is for totipotent stem cell. It will give rise to pluripotent stem cell. It will give rise to multipotent stem cell, and they will further differentiate into fully differentiated cells or the unipotent stem cells. See here the rota is there for the rubrics are here for the color coding. See, this color, greenish color, is for totipotent. Totipotent will give rise to pluripotent. Pluripotent will give rise to multipotent, and multipotent will give rise to unipotent cell. Okay, so this is it. Now the next topic of importance is the innovations in industries. See, industries are using one important thing. Generally they are using the enzymes. In every industry enzymes are being used, so enzyme play a very important role when we talk about industries. So the first thing I'll be discussing here is the concept of enzyme engineering. Just like genetic engineering, when we alter the genes in such a way that the enzymes which are produced from those genes are better than the naturally produced enzyme, this whole methodology of changing the gene is called as enzyme engineering. Okay, it is actually a type of genetic engineering where new variety of enzyme can be produced by altering the gene responsible for the production of that particular enzyme. Okay, so what will happen once the enzyme engineering is being done? The new enzymes will be produced, and then the study of structural features related to change and production of stable enzymes produced can also be studied, that how a more stable form, maybe heat resistant, maybe pH resistant enzymes can be created. So such type of innovations are taking place, are being done in enzyme engineering. Okay, the most exciting development over the last few years is the application of genetic engineering techniques to enzyme technology. This I just now told you. The amount of enzyme produced by a microorganisms may be increased by increasing the number of gene copies that code for it. If we know that a particular gene codes for a particular enzyme and we have recognize that particular gene, if we engineer the gene in such a way that that particular gene is being repeated after certain duration of time, that is after a short stretch of DNA, the gene which is coding for the enzyme is again repeated, and then after short stretch it is again repeated. So what we will have, we will have the enzyme being produced in a larger quantity as it was being produced earlier. Okay, so that is a very beneficial point of enzyme engineering. Large amount of enzymes can be created. Okay, moving forward, there are different utilizations, different uses of enzymes technology. Enzyme can be used as biocatalysis, can be used in research and development, can be used in diagnostics, can be used in food and beverages, can be used in animal feed, can be used as in pharmaceutical industries. Enzymes can be used as biofuels, and they can be used as household items for cleaning agents, etc. Now we will study about certain applications of enzymes. The first application of enzyme is the therapeutic use or the medical purpose, that is in the treatment of some medical conditions. Here what will happen, if some patient, some person is suffering from a disease where the enzymes which are required in the body are not produced, then those enzymes can be supplied from outside. Such kind of enzymes are being studied under therapeutical uses. Examples will be anticoagulants or lactose intolerance. There are enzymes which are responsible for lactose tolerating inside the body, and there is no ill effect or allergic reaction from the body when the lactose is being eaten up. So what happens in them when we are introducing certain enzymes in their body? When the lactose intolerance enzyme is not produced in the body, these enzymes can be supplied from outside, so that the person can develop lactose tolerance. They are no more intolerant to lactose. Similarly, the next use is an analytical use of enzymes like lipase. Lipase will be used in triglycerides estimation. Taq polymerase is a very important enzyme which is used in analytical purpose for replication of DNA, making copies of DNA. So in analytical uses also enzymes are being utilized. Next is industries industrial use, such as dairy products. The rennet enzymes, calf rennet, can be used to curdle the milk to make cheese in cheese industries. Rennet is a very important enzyme which is used in the production of cheese. Okay, so it is very important, you need to remember the examples. Second is in laundry, the enzyme which is used is subtilisin, which is used to are either which are used in normal washing, which are used in washing purpose, so that what will happen, the proteins which are stuck on the fibers can be removed easily by this subtilisin enzyme, which is a bacterial origin. Next is food industries. Papain can be used. Papain is again a proteolytic enzyme which can be, as the name says, it is, we are getting it from papaya. So papain enzyme is effectively working on the meat and protein of the meat, and is softening the protein or tenderizing the meat so that it is easily being chewed and can be eaten easily. One more you have to study is the use of enzyme in the brewing industry. The alpha and beta amylases which are used in brewing industry to produce, brewing industry, remember I have told you in class 11th, beer production and other alcoholic beverages, to enhance the taste. These kind of alpha and beta amylase are used. Also this amylase enzyme can be used in the bakery in the process of bread making. So there is a table also given in your book at page number 75, industrial enzyme. Just go through that page because it is very important, and these any enzyme can be asked. Mostly these enzymes which I have covered, the anticoagulants, lactose intolerance, lipase, Taq polymerase, rennet, subtilisin, papain, alpha beta amylase are generally asked. But then also you go through page number 75, and here is a table: industry, leather tanning, starch processing, detergent, textile, chemicals, food processing, biofuels. In environmental, enzymes used for residual, for degradation of the waste, for detoxification of toxic substances. These things you don't have to study in detail, but just keep it into your mind that in agriculture also the enzymes can be used to animal feed, as an animal feed additive. Food processing and agro processing, these things have to be kept in mind. They are not specified in your syllabus, but then also you can learn them. Okay, so we stop here. Today we have completed the innovations chapter. Just go through the chapter. I'll be sharing with you the notes soon so that you can copy them or paste it in your notebooks. Hope you enjoyed the chapter, and start reading, find out your own notes, find out your own literature, and start reading. Thank you so much.